Welcome, my dear students and other YouTube viewers, to this chapter in biochemistry in which I will teach you digestion, how to turn meals into cellular biochemicals. Before we begin, we always like to start with something hilarious. Instead of showing you a chemistry cat for today, I opted to share something funny from howtobeadad.com that my wife emailed me some time ago. Some of you with kids might have experienced a few of these baby sleep positions. My favorite one, though, of all of these, because I've experienced this back when my kids were little, is H is for hell. Ha ha ha! We now move on then to an interesting molecule of the day. The one I'm featuring today is delta-9 tetrahydrocannabinol, also known as THC. It happens to be the psychoactive compound in marijuana or cannabis sativa. It was first isolated by Y. Ganoi and R. McChollum in 1964. THC receptor CB1 in the brain gives the marijuana high, while receptor CB2 in the immune system is the basis for its medicinal use. From 1984 to 2006, J.W. Huffman and coworkers used THC and its analogs in immunosuppressant drug research. Isn't that interesting? Good. With that said, after this chapter, you should be able to do the following. First, know the 3 stages of digestion. Second, thoroughly describe how proteins, carbohydrates, and lipids are each digestively broken down. And third, describe generally the way in which signal transduction induces feelings of satiation or fullness, also called satiety. Lot to get to, so let's get started. Right here with digestion happening in 3 stages. So as it turns out, when we eat food, our digestive systems break it down into smaller molecules that can then be used as building blocks for metabolism. Digestion occurs then in 3 stages. 1. Large molecules are broken down into smaller units. 2. The resulting small molecules are broken down even further. And 3. ATP is produced by oxidizing acetyl-CoA. These 3 phases of digestion are broken down into these 3 little rectangular regions in this figure from our book. Now, step 3, by the way, is called the citric acid cycle, Krebs cycle, or sometimes TCA, which stands for tricarboxylic acid cycle. And we will spend a lot of chapters discussing each of these phases in excruciating detail. As an additional note, ATP, adenosine triphosphate, has this structure, and you might recognize this nucleotide base as adenine, the A that we find in DNA and RNA. By analogy, acetyl-CoA has a similar or related structure. You can see that over here on this side, it resembles ATP, except that it's adenosine diphosphate. It only has 2 phosphate groups over here, and then a third attached to this hydroxyl at this position. And then it has all this extra structural stuff over here on this location. Where does the term acetyl come from? Well, as it turns out, the word acetyl refers to this specific structure right here. Carbon-oxygen double bond flanked by a CH3. So that tiny little appendage dangling off of the sulfur is an acetyl group. And as small as it is compared to the rest of the molecule, that's where the term acetyl comes from in acetyl-CoA. In this chapter, then, we will address a fundamental mystery that has plagued and intrigued deep thinkers since the beginning. How in the world does food turn into poo? Now, to properly cover digestion, we'll need to learn the following 3 things: how proteins are broken down, how carbs are broken down, and how lipids or fats are broken down. Are you ready for this? Please buckle up. Let's get into it. Starting with how digestion begins. So when we eat food, the following process begins. First, we homogenize or puree our food by chewing. Second, stomach acid with a very low acidic pH denatures or breaks apart the proteins in our food. Third, the proteolytic or protein-eating enzyme pepsin breaks these proteins down into smaller fragments called oligopeptides. Fourth, the gallbladder, if you're lucky enough to have a gallbladder. See, I don't. I'm actually a gallbladderless freak because I had to have my gallbladder removed years ago because of some health problems. Anyway, but the point is, if you have a gallbladder, your gallbladder releases bile salts into the stomach, which then aid in breaking down lipids. Fifth, as broken down molecules begin to enter the intestine, the pancreas also stimulates the release of sodium bicarbonate, a base, to neutralize the pH of the food as it leaves the stomach, so that that low acidic pH in the stomach remains more or less localized to the stomach, ideally. Baking soda, by the way, is sodium bicarbonate. And sixth, the pancreas releases an army of digestive enzymes. So this is the beginning of digestion. Let's get into some details. So as I just mentioned, once proteins are degraded by our stomach acid, which is HCl or hydrochloric acid, the enzyme pepsin begins to break them apart into smaller peptides called oligopeptides. Pepsin, by the way, is a protease. Proteases are also called proteolytic enzymes, which are enzymes that break apart proteins into individual amino acids through the following reaction called hydrolysis. In this reaction, whose mechanism I won't show in detail here, but I will link to a video above my head here and in the description below to review this from organic chemistry if you want. The oxygen in H2O is brought in, it forms a bond with this carbon, pushes pi electrons up onto this oxygen, then slaps pi electrons back down and severs this carbon-nitrogen bond, kicking this off. There's a hydrogen swap exchange in which you end up with the OH on this position that came from your H2O getting deprotonated, so you ultimately end up with a carboxylate at this position, and then you end up with a protonated amine or ammonium at physiological pH. In effect, this step, hydrolysis, shaves apart or breaks apart the nitrogen-carbon bond, the amide bond or peptide bond at this location of the protein. As pepsin catalyzes this reaction in many, many different locations across a protein, it breaks it down into smaller and smaller individual peptides, also called oligopeptides. Make sense? Good. As you may recall, back in chapters 5 and 6, we learned about 3 proteases: papain, trypsin, which cleaves peptide bonds on the carboxylic acid side of lysine and arginine, and chymotrypsin, which cleaves peptide bonds on the carboxylic acid side of phenylalanine, tyrosine, and tryptophan. So once pepsin has begun breaking down proteins in our stomachs, the peptides, carbohydrates, and lipids from our food are moved into the small intestine, where a hormone called secretin is released by the pancreas to stimulate secretion of sodium bicarbonate. This helps neutralize the pH of the stuff as it exits the stomach. At this stage, a hormone called cholecystokinin, or CCK, prompts the pancreas to release its army of digestive enzymes into the lower intestine to further aid the digestion process. Now, the enzymes that get released by the pancreas are actually in inactive form initially. Inactive enzymes are called proenzymes or zymogens, and for some reason I personally dislike the word zymogen. I don't really know why, so I'll just be using the term proenzyme instead, okay? Now, at the entrance to the small intestine, an enzyme called enteropeptidase activates the proenzyme trypsinogen, which converts into its active form, trypsin. Does that sound familiar? Of course, I just talked about trypsin and chymotrypsin a moment ago. Trypsin then activates all of the other proenzymes, the army of enzymes. Got it? Good. All right, so once deployed and activated, this enzyme army, aided by a final enzyme called peptidase, hydrolyzes proteins into individual amino acids and di or tripeptides. These amino acids and di or tripeptides are then released through the intestine wall and into the blood, where they can be picked up and used by other cells all over our bodies. The process of protein breakdown then is summarized by the figure shown here, which I invite you to pause and take a look at. We end this video, the first of 3 on this chapter, with a quick quiz. By looking at the figure shown here, please explain why a person with a trypsinogen deficiency would suffer from more digestion difficulties than a person lacking most of the other proenzymes. Now, I'm not going to answer this here, but I invite you to try it on your own. Then, once you're done, you're welcome to look up the answer on page 245 of the textbook for this class. Thanks a lot, my dear students, for tuning in. Please tune into the next video in which we'll continue this chapter's coverage of digestion.